Welcome to the deep dive. You know, for decades, when we look at really complex diseases, things like cancer or severe inflammation, we've basically relied on maps.
Spatial maps?
Exactly. Beautiful, highly detailed spatial maps. They show us every single cell, what type it is, and exactly where it's sitting in diseased tissue. But, well, mapping is descriptive, right? It just tells us what happened.
And that's precisely the leap we're talking about today. Imagine going beyond that static map to having a tool that's predictive, even generative.
Yes.
Like a simulation.
Okay.
We're diving into this really fascinating AI algorithm called MintFlow. And the idea here is it's not just analyzing the tissue map, but it's designed to potentially actively reprogram those tissue microenvironments right there in the computer. It lets us run basically virtual clinical trials.
Virtual clinical trials, that's a big claim.
Yes.
So what's the core problem it's trying to solve?
Well, it tackles something fundamental but really tricky in biology. When you look at a cell and measure its gene expression, what it's actually doing at that moment. How much of that activity comes from the cell's own basic identity, its intrinsic programming, versus, well, how much is it just reacting to its neighbors?
Like cellular peer pressure.
Exactly like peer pressure. Current computational methods, they tend to struggle to pull those two things apart. They just give you the combined signal.
Right, the final outcome.
Yes. And MintFlow's big innovation is that it uses AI to mathematically isolate that intrinsic signal from the effects induced purely by the microenvironment. And that separation is what unlocks the power to simulate tissue changes, perturbations with real predictive ability.
Okay, let's unpack this core mechanism then, because this sounds like where the real AI magic is happening. You mentioned it's built on something called flow matching. What does that mean for us conceptually? Not the deep math, but the idea.
Right, okay. So think about flow matching, like calculating the precise path or the flow between two states. We have the cell's sort of baseline, intrinsic state, and we have the final state, which is the gene expression we actually measure.
Okay.
Flow matching helps figure out the complex transformation that happens when the environment signal gets added to that intrinsic state. It gives us a mathematically sound way to calculate the environment's exact influence, and crucially, it does this without needing prior knowledge, like guessing which specific molecules are talking to each other.
So it learns the communication rules from the data itself. What data does it actually need to perform this calculation? You need the cell, its type, and its neighborhood, right?
That's spot on. MintFlow treats the incoming and outgoing communication signals as what we call latent variables, these hidden drivers. So for every single cell on that map, it needs three key inputs.
Okay, what are they?
1, the cell's total measured gene expression. 2, its basic cell type label, the T cell, a fibroblast, a cancer cell. And 3, this is critical, the microenvironment cell type composition, or MCC.
MCC, that sounds like the local census report you mentioned.
It basically is. It's a precise snapshot of all the different cell types within that cell's immediate vicinity. Technically, the paper uses the 5 closest neighbors based on a spatial graph.
Got it.
And with those 3 pieces of information, the algorithm learns how to split that total gene expression signal into 2 really distinct components.
Okay, 2 buckets. Let's call them bucket 1.
Bucket 1 we can think of as the self-driven component. The paper calls it A-Day. This is the expression driven by the cell's own internal machinery. Things like receptors it already has, maybe its cell cycle state. Its inherent personality, if you like.
Its personality. Okay, and bucket 2.
Bucket 2 is the environment-induced component. This part of the expression is driven purely by that spatial context, by the signals it's getting from those neighbors we just counted in the MCC. This is the peer pressure effect quantified.
That separation is incredibly powerful conceptually. So once you have these 2 separated components for every cell, what can you actually do with that information? What are the outputs?
This is where it gets really useful. Once that signal is split, you get a few key things. First, a microenvironment score for each cell. It quantifies exactly how much the environment is influencing that specific cell. This immediately highlights spatial signaling hotspots in the tissue.
Okay, zones of high influence. What else?
Second, you get the microenvironment-induced gene programs, or MGPs. These are the actual lists of genes that are turned on or off, specifically because of those external cues from neighbors. And you get this at single-cell resolution.
And this detail, the separation, is what enables the simulation part, the in silico perturbation.
Exactly, that's the big payoff. Because MintFlow has learned the rules of how the environment influences cells in that specific context, you can start simulating changes. You can ask the computer, okay, what if we performed a virtual cell deletion? Let's remove all the macrophages in this region.
It replace cells.
Or cell replacement, yeah. What if we change these inflammatory T cells into regulatory T cells? And MintFlow can then predict the resulting change in gene expression in all the neighboring cells. This ability to test hypotheses virtually at scale is just transformative.
But hold on, how do we know these predictions are, well, accurate? If you virtually delete a cell, maybe the model's prediction is just a guess based on correlations, not causation. How is this validated?
That's a crucial question. The validation was twofold. First, they ran standard benchmark simulations comparing MintFlow to other existing methods, and it significantly outperformed them in correctly separating these signals.
Okay, benchmark tests.
But maybe more convincingly, when they applied it to real tissue data, MintFlow consistently assigned a much larger fraction of the expression of known signaling genes, things like cytokines and chemokines, which we know are involved in cell communication. It assigned those to the environment-induced component.
Because logically, those communication genes should be driven by the environment.
Precisely. So the fact that MintFlow naturally puts them in that bucket provides strong evidence that it's correctly isolating the extrinsic, the environmental influence.
That makes a lot of sense. It's a really clever internal check. Okay, so MintFlow seems to be a validated detective for figuring out who's influencing whom in the tissue. Let's see it in action. You mentioned 3 case studies. Where do we start? Inflammation.
Let's start with atopic dermatitis, or AD. It's a really common chronic inflammatory skin disease, often driven by what's called type 2 inflammation. Think T cells, cytokines like IL-4, IL-13, causing itchy, inflamed skin.
Right. So what did MintFlow find there? Did it pick up on the inflammation?
Immediately. When they calculated the microenvironment scores across the skin samples, the high scores lit up right in the superficial dermis. And when they compared this map to standard histology slides, those high-score regions perfectly matched the areas where pathologists see dense clusters of inflammatory cells around blood vessels, the perivascular infiltrates.
So it pinpointed the physical location of the inflammation hotspots. What about the nature of the signaling there? What did the MGP, the environment-induced gene program, reveal about that specific zone?
Well, when they clustered cells based purely on their environment-induced genes, a specific microenvironment signature emerged, which they called the TDC domain. This domain was much more prominent in the inflamed AD skin compared to healthy skin.
TDC, T cells, and dendritic cells.
Exactly. It was mainly composed of T cells and various antigen-presenting cells, particularly a type called migratory dendritic cells, or MGPs. The analysis confirmed this TDC domain was functioning essentially as a T-cell activation hub.
And what specific activities defined that hub? What kind of molecular conversations were happening?
The gene program for that domain, the MGP, pointed clearly to key processes, things like T-cell activation markers, genes involved in T-cells sticking to APCs for communication, like ICM3, and also genes pushing the immune response towards that type 2 profile, like ALOX15.
So, confirming the known biology but with more detail.
Yes, and it also identified specific communication axes. It confirmed a known pathway, CCL19 ccR7, which often involves fibroblasts recruiting immune cells. But it also highlighted another axis, CCL22 cc17 interacting with ccR4, as being really important for the MGDC T-cell interactions within that hub.
Okay, so a very detailed picture of the known inflammatory process. But didn't MintFlow also uncover something new, something missed by standard analysis?
It did, and this is really where the power of disentangling these signals shines. By looking at the intrinsic T-cell programs, after removing the environmental noise, MintFlow found a distinct T-cell cluster that wasn't obvious before.
What kind of cluster?
It was a population of CD8A plus T cells that also expressed specific markers like ITGAE, GZMB, and importantly, the type 2 cytokine IL-13. Given its specific location in the superficial dermis and this unique marker profile, they characterized it as a novel, spatially defined type 2T resident memory cell, TRM population, likely specific to the AD skin environment.
Wow, so a new cell state revealed just by separating out the environmental shouting. That's impressive. Now, let's get to those virtual experiments in the AD model. They focused on regulatory T cells, TREGs, right? The immune suppressors.
Yes, TREGs are crucial for keeping inflammation in check.
So perturbation 1, virtual TREG depletion. What happened when they digitally removed TREGs from that TDC activation hub?
The prediction was quite stark. The neighboring MGDCs, those key antigen-presenting cells, significantly ramped up their expression of inflammatory genes. Things like the chemokines CCL17 and CCL22, which attract more inflammatory cells, and pathways involved in inflammation signaling like MAPK cascade regulation.
So the prediction was removing tregs leads
It's
to
increased inflammation,
say it.
a worsening of the AD-like state in the simulations.
And that prediction actually lines up with real-world human biology, doesn't it?
It does remarkably well. There are rare but severe genetic conditions like IPEC syndrome, where patients have dysfunctional tregs and they suffer from extreme allergic inflammation, including severe AD. So the simulation matched known clinical outcomes.
Okay, that's strong validation. What about the opposite experiment? Perturbation 2. Treg augmentation. What if they digitally added tregs?
When they simulated replacing other T cells with tregs in that hub, those inflammatory pathways were not enriched. In fact, the overall prediction was an anti-inflammatory effect, a quieting down of the hub.
And was there clinical data to support that prediction too?
Yes, indirectly. They linked this to in vivo data from patients treated with dupilumab, an effective AD drug. Clinical responders to the drug often showed an increase in treg numbers in their skin after 12 weeks of treatment, suggesting that having more tregs is indeed associated with therapeutic benefit, just as MintFlow predicted.
Okay, so MintFlow accurately modeled the impact of tregs in AD inflammation. That's powerful. Let's switch gears now. Inflammation involves a lot of chemical signaling, but what about cancer, where the physical structure, the architecture of the tumor environment is often a huge barrier? How did MintFlow handle cutaneous melanoma?
Right, melanoma is a classic example where the tumor microenvironment, the TME, plays a huge role. And interestingly, when MintFlow calculated the microenvironment scores across the melanoma samples, the highest scores, the areas with the most intense environmental signaling, weren't actually inside the nests of tumor cells.
Really? Where were they?
They were predominantly outside the tumor nests, in the surrounding structural tissue, the stroma.
So the most intense cellular crosstalk was happening at the tumor border, in the supporting tissue, not deep within the cancer itself.
Exactly. So MintFlow zoomed in on this stroma domain, which is mostly made up of cancer-associated fibroblasts, or CCAFs, and also contains immune cells like T cells. And the MGP, the environment-induced gene program for that specific stromal domain, revealed a really sophisticated two-part mechanism that actively excludes T cells from getting to the tumor.
A two-part barrier. What was the first part? How are T cells kept out?
The first element was chemotactic exclusion. The MGP showed very high expression of a specific chemoattractant molecule called ccRCC.
ccRCC, I know that one. It interacts with CXCR4 on T cells, right?
That's right. And this ccRCC-CXCR4 axis is well known in cancer biology for basically luring T cells away from tumor nests. The MintFlow data showed T cells congregating in these high ccRCC stromal areas, effectively being kept at a distance from the tumor cells they're supposed to kill.
So they're being actively baited away by the stroma. What was the second part of the barrier? You said physical structure.
Yes, the second element was a physical barrier. The stroma MGP was heavily enriched for genes that build the tissue scaffold, the extracellular matrix, or ECM. Specifically, lots of collagen genes like CO4A2, CO5A1, COL1, which make the tissue dense.
Okay, dense collagen.
But crucially, it wasn't just the collagen itself. The MGP also showed high expression of enzymes like LOX, LOXL1, and LOXL2. These enzymes cross-link the collagen fibers.
Ah, making it stiffer and harder to penetrate.
Exactly. Increased collagen cross-linking makes the stroma incredibly dense and stiff, creating a literal physical wall that T cells struggle to get through. It's associated with worse outcomes in many cancers.
Okay, so a chemical lure and a physical wall built by the stroma. But then MintFlow made an even more surprising connection, linking this melanoma finding to a completely different condition.
Yes, this was fascinating. They took that specific stroma MGP signature from the melanoma, that program of CXCL12, collagens, and cross-linking enzymes, and they scored it across data from 23 different skin diseases.
And where did it show up strongest?
It showed the highest expression, unsurprisingly, in melanoma. But the second highest hit was in keloid scarring.
Keloids, those really thick raised scars.
Exactly. Keloids are a type of fibroproliferative scar characterized by excessive deposition of dense, highly cross-linked collagen. So MintFlow essentially uncovered that the melanoma TME induces this inducible keloid-like stroma. It's borrowing a wound-healing scarring program to build a barrier that acts both physically and chemically to keep T cells out, creating a safe haven for the tumor.
That's a really striking finding, a tumor co-opting a scarring mechanism. Okay, final case study. Clear cell renal cell carcinoma, ccRCC, kidney cancer. This one gets into complex tissue structures and links directly to patient survival, right?
That's right. ccRCC is known for its complex architecture. Tumors often contain these large organized immune cell aggregates called tertiary lymphoid structures, or TLSs. Their presence is actually linked to how well patients do.
Okay. So did MintFlow find these TLSs?
It did. When they applied MintFlow to ccRCC spatial data, the microenvironment score clearly highlighted specific hotspots of intense signaling. In one particular tumor core, this hotspot perfectly corresponded to what pathologists confirmed, under the microscope, was a well-formed TLS.
So it identified the structure. But TLSs are notoriously complex, packed with different immune cells. Could MintFlow see inside the TLS with more detail than usual methods, especially the T cells?
Absolutely. Standard analysis often just lumps T cells into TLS together. But MintFlow, using its ability to look at the environment-induced states, was able to resolve much finer detail. It identified 6 distinct T cell states within the ccRCC samples.
6 states instead of just T cell.
Yes, and importantly, 3 of these states were uniquely found within the TLS structure. They called them TLS Core 1, Core 2, and critically, the TLS border T cells.
TLS border T cells. What was special about that population sitting right at the edge of the structure?
They were clearly immunosuppressed. Their gene expression was highly enriched for classic T-cell exhaustion markers, genes like HAVCR2, which encodes TOXLAG3, cXCR3. These cells were basically shut down, functionally exhausted, right there at the boundary of this potentially anti-tumor structure.
Okay, so exhausted T cells at the border. The obvious question then is who is exhausting them? Which cells in that dense TLS niche are sending the shutdown signals?
Well, standard ligand receptor interaction predictions pointed towards myeloid cells, and specifically macrophages, as the most likely culprits interacting with these TLS T cells.
Right.
But MintFlow went further. By subclustering based on the environment-induced signals, it confirmed the existence of a distinct TLS-associated macrophage subset. These macrophages express specific markers like TREM2 and CCR1, and were physically localized right there within the TLS cheek by jowl with the exhausted T cells. Their gene profile was strongly linked to T-cell co-inhibition.
So MintFlow nailed down not just that macrophages were likely involved, but identified a specific subtype of macrophage sitting in the right place and expressing the right inhibitory molecules.
Exactly. It implicated these specific TLS-associated macrophages as the dominant suppressors driving T-cell exhaustion within that niche.
This sounds like the perfect scenario for one of those in silico perturbations. What happened when they simulated getting rid of these suppressive macrophages?
They did exactly that. They performed a virtual deletion of just those TLS-associated macrophages within the simulation.
And the predicted effect on the neighboring T cells, did they wake up?
The predicted effect was consistent and really quite striking. The remaining T cells, particularly those TLS border T cells, showed immediate signs of reprogramming. They started downregulating those exhaustion and co-inhibitory markers, things like PDL2, TIMM3, HAVCR2, LAR1, VSIR.
So the off signals decreased. Did the on signals increase?
Yes. Simultaneously, they showed increased expression of genes associated with T-cell activation and function, like PLCG1 and IL4R. The simulation predicted that removing those specific macrophages effectively restored the T cells towards a more active functional state.
Okay, that's a powerful prediction. But the ultimate test is, does this virtual reprogramming have any connection to actual patient outcomes? Does this predicted waking up of T cells matter clinically?
This is maybe the most compelling part. They connected these findings directly to real-world patient survival data from the Cancer Genome Atlas, the TCGA cohort, which included over 600 ccRCC patients.
And what was the link?
They looked at the gene expression programs. High expression of the original TLS border T-cell program, that exhausted, immunosuppressed state identified by MintFlow, was significantly associated with worse overall survival for patients, p equals 0.0073.
Okay, so having those exhausted cells is bad news. What about the reprogrammed state?
Here's the kicker. High expression of the reprogrammed T-cell state, the predicted profile after the virtual macrophage removal, was associated with a significant survival advantage. P equals 0.0034.
Wow. Let me make sure I understand this. MintFlow identified an exhausted cell state linked to poor survival. It predicted how to fix it, remove specific macrophages, and the predicted fixed state is actually linked to better survival in real patients.
That's precisely it. It connects the dots from observing a structure, the TLS, to identifying its specific cellular dysfunction, exhausted T cells due to specific macrophages, to predicting a targeted intervention, macrophage removal, and then validates that prediction by linking both the broken and fixed states directly to patient survival outcomes in a large data set.
That is genuinely staggering. It's moving way beyond just mapping.
It really is. It fundamentally changes how we can think about identifying therapeutic targets. MintFlow isn't just describing the tissue landscape, it's providing a predictive model that lets you simulate rewriting it. It allows researchers to perform these really complex experiments, like precisely deleting one cell type or replacing it with another, experiments that are currently incredibly challenging, expensive, or frankly, often impossible to do at scale in a wet lab.
Okay, let's try to distill the key takeaways from this deep dive into MintFlow. What are the main messages we should be taking away?
I think there are three core points. First, MintFlow's ability to separate intrinsic from environment-induced signals is key. It uncovers clinically relevant biological processes and cell states that were previously hidden in the noise, like that keloid-like stroma in melanoma, or the novel type 2 TRM cells in atopic dermatitis.
Right. Finding the hidden biology? What's the second point?
Second, it directly links that microenvironment-driven pathology to actual clinical outcomes. The example of the TLS border T cells in kidney cancer predicting poor survival, and the reprogrammed state predicting better survival, is a prime example. It makes the findings translationally relevant.
Connects the mechanism to the patient. And third,
And third, it dramatically accelerates hypothesis generation for new therapies by providing highly specific, context-aware targets. Instead of just saying target macrophages, it says target these specific TLS-associated macrophages expressing these markers because they're suppressing T cells in this way. Or consider modulating trigs in AD because we predict this specific effect on MGPs. It allows for much more focused translational research.
it really feels like we're witnessing a fundamental step change here, moving towards something like AI-driven construction of virtual tissue models. I mean, if you can accurately simulate disease progression and even predict the response to targeted interventions entirely within a computer, how quickly could that shift drug discovery and therapeutic design away from broad trial and error towards truly context-aware, personalized precision medicine?
It suggests the tissue microenvironment isn't just a passive backdrop anymore. It's an active, dynamic system that we might be able to learn how to program or at least nudge in beneficial directions.
Yeah.
Implications are pretty profound.
Absolutely. It makes you wonder what other incredibly complex tissue interactions in diseases we haven't even touched on today are just waiting to be unraveled by tools like MintFlow. Tools that can finally look beyond simple descriptions and start predicting the dynamic biology underneath. That's definitely something to think about for the future of medicine. Until the next deep dive, keep learning.